Welcome to The Explainer. Today we're diving into a super fast quiz all about genetic interactions. This is where the simple rules of heredity get, well, really interesting and complex. You think you're ready to play?
Okay, it's time to put that brain to work. We are going to move quick, so try to keep up. Can you tell your alleles from your epistasis? Let's find out. Let's do this!
All right, here's how it's going to work. Super simple. I'll throw a question at you, you get 5 seconds to think about it, and then we'll dive into the answer together. Let's kick things off.
All right, round one is called decoding dominance. We're going to start with the basics, the fundamental rules of how different versions of a gene, we call those alleles, make themselves known.
Okay, first question's on the board. What is complete dominance? Your 5 seconds starts now.
So, what'd you get? Complete dominance is basically when one allele, the dominant one, just completely masks the other, the recessive one. Think about it like this. If a cat has just one dominant allele for a black coat, guess what? The cat's going to be black. The white coat gene might still be hanging around in its DNA, but it's totally silent.
Okay, next question. How are incomplete dominance and codominance different? They sound kind of the same, I know, but trust me, they're not. 5 seconds. Go for it.
All right, let's clear this up. Incomplete dominance is all about blending. It's like mixing paint. You take a red flower and a white flower and boom, you get pink flowers. But codominance, that's different. Both traits show up fully and separately. The classic example is AB blood type. You don't get some weird blend of A and B, you literally have both A and B antigens on your cells. So think blend versus both.
Okay, round one is done. Now for round two, when genes interfere. This is where things get really cool, because we're talking about genes at totally different spots on the chromosome that start messing with each other.
All right, this is a huge concept in genetics, so focus up. What exactly is epistasis? You know the drill. 5 seconds.
So epistasis is when one gene basically has the power to shut down or change what another gene does, even if that other gene is somewhere else entirely. In maize, for instance, the RR genotype is like a kill switch. It doesn't matter if the gene for purple color is there. If RR is present, the grain is white. It completely silences the color gene. That's the key takeaway. One gene can tell another gene to be quiet.
Okay, let's get a bit more specific. What happens when it's the dominant allele that's doing all the masking? What do we call that? That's dominant epistasis. You've got 5 seconds.
And that is dominant epistasis. A great example is in summer squash. If a squash plant has that dominant W allele, it's going to be white, period. It doesn't matter one bit if it also has the gene for yellow fruit. The W allele is the boss, it just overrides everything else.
Ready for the next one? Okay, what are duplicate genes? Take 5 seconds.
Think of duplicate genes as having a backup system. In the shepherd's purse plant, they are 2 different genes that can do the same job. As long as you have a dominant version of either gene A or gene B, you get a triangular seed pod. The only, and I mean only way to get an oval pod is if you have the recessive versions of both, AABB. It's like having 2 chances to get it right. All right, last question for this round. What is an inhibitory factor in gene expression? What does an inhibitor gene do? 5 seconds. Okay, an inhibitor gene is exactly what it sounds like. It's a blocker. By itself, it doesn't really have an effect. But when you have the dominant version, like the I gene in rice, its whole job is to stop another gene from working. In this case, it stops the P gene from turning the leaves purple. So even if the plant has the gene for purple, the inhibitor just says, nope. And here we are, our final round. One gene, many effects. Now we're looking at single genes that don't just do one thing, they have these massive, widespread impacts on an entire organism.
This next one is a really powerful idea in genetics. The question is, what is pleiotropy? You've got 5 seconds.
Pleiotropy. It's when a single gene pulls double, triple, or even quadruple duty. It influences multiple traits that might seem totally unrelated. The most well-known and pretty serious example is sickle cell anemia. A mutation in just one gene doesn't just affect red blood cells. It triggers this whole domino effect of issues, like intense pain, organ damage, and a bunch of other symptoms. It's a stark reminder of just how connected everything in our body really is.
Okay, final question. Let's step back from the textbook definitions for a second. What are the real-world, human implications of genetic disorders like sickle cell anemia or PKU?
Let's think about that for 5 seconds. The impact is just huge. It's not just about biology. It's about people's lives. We're talking about chronic health problems, daily struggles, like the incredibly strict diet someone with PKU has to follow. And then there's the emotional toll, the financial burden on families. It's so important to remember that these aren't just terms in a quiz. They are the daily reality for millions of people.
And that's it. We're done. How did you do? Hope you kept up. Before we go, let's just do a lightning-fast recap of the big ideas we hit today.
We went from complete dominance to codominance. We wrestled with the complexities of epistasis, we saw how duplicate genes work as backups, and we finished with the massive reach of pleiotropy. Not bad for a quick quiz, huh?
You know, if you take one thing away from all this, it should be that genetics is so much more than a simple instruction manual. It's more like a complex, beautiful, and sometimes chaotic dance between genes. Every time we think we have it figured out, we discover a new layer of complexity, which really makes you wonder, right? What other incredible genetic secrets are out there, just waiting to be discovered? Thanks for playing along today.